Hello everyone, welcome back to Chinmoy's Biology channel. So let's get started with a new episode of Heredity. So it is part 2 and we have already completed till the monohybrid cross. Today we are going to start from the Punnett square. So this is what we have studied in theory in the last part. So please refer to that part if you have not yet gone through that. I'll be putting that link in the description box below. Now the parents were pure tall, one was pure tall and one was pure dwarf. So I told you that we are writing the dominant characters in the capital form and the recessive characters in the small form. But these are all homozygous here. So this is also homozygous, this is also homozygous, which means that both the alleles are here capital T and both the alleles are here small t. Now what all gametes we are getting? So one from this is one capital T from this and one small t from this. Now the heterozygous tall plant in F1 generation, we are getting all the plants which are heterozygous tall. So all the plants in F1 generation are tall. So then what was done? Then Mendel did selfing. So what is selfing? Selfing is he crossed this plant again with the same TT. So this selfing was done and this is the Punnett square. So what is Punnett square? We are writing the female gametes on one side and we are writing the male gametes on one side. So like that we will do this cross. So T 1 from each we are taking. So T into T. So 1 TT is there. Then T into T, capital T small t has come, then again small t into capital T, so capital T small t is there, then again two small t's are there, TT, a dwarf plant is there. So what is the phenotypic ratio here? What is visible here? So visibly we will see this and this three are tall plants and this is a dwarf one. So the phenotypic ratio is 3:1. Okay, so phenotypic ratio is 3:1. Whereas what about the genotype? So the genotypic ratio we can see this one, it is absolutely, it is a pure tall plant. Okay, so this is a pure tall plant. So 1, then this and this, these are heterozygous tall, so is to 2, and then this one is a pure dwarf plant, is to 1. So is it clear to you all? So the phenotypic ratio is 3:1 and the genotypic ratio is 1:2:1. Now it is the Punnett square. Now we will move on to the NCERT book where we will discuss what they have told you and we will move on to the dihybrid cross with the Punnett square. So don't skip my lesson in between, because if you are skipping you will miss many important points and topics which are being taught throughout the episode. So stay tuned, and if you are liking my channel, obviously please do subscribe to my channel. Stay tuned, stay updated. Okay, so let's get started with that part. So from where we are getting the concept of gene, we should know that. So inside the cell there is nucleus. Inside the nucleus there are chromosomes. Okay. So these chromosomes are having DNA inside them, and one part of the DNA which is carrying the code, or which is carrying the characters or traits, is the gene. Okay. So DNA are double helical structure. So this details in higher classes in 12th and all we will study. They are made up of adenine, thymine, cytosine, guanine. These are nucleotides. With this nucleotide base pairing only the DNA is formed. Okay, and this is a sugar phosphate backbone. This helix is made up of a sugar phosphate backbone, and these are all nucleotide bases. Okay, so let us move on to, as per your NCERT book, what they are telling. So now till now the concept of gene is clear to you all now. Okay. So here we come with the dihybrid cross. Here, as per Mendel, we are taking pure breeding plants, that is pure breeding yellow and round seeds are RR, YY, and pure breeding wrinkled and green seeds are small RR and small YY. Okay, so now the gametes are this RY and RY from these two parents. Now in the F1 progeny or F1 generation, the first filial generation, we are getting all round and yellow seeds. Okay, is it clear? But in F2 generation, when we are doing selfing, what is selfing? I have told you earlier that it is a cross between the same type of plants. So they are crossing RRYY with another set of RRYY itself. So no different plants is there. So here they are taking two pairs of contrasting characters because here the similar type of plants are being crossed here. So another RRYY is being crossed, and that is self-pollination. Okay. Now what we are getting? We are getting these combination of gametes from these two plants. So the gametes are RY, capital R, capital Y, small r, capital Y, capital R, small y, and small r, small y. So this is the gamete combination. Okay. So here if we are writing the male gametes here, and here we are writing the female gametes, so the Punnett square will look as this. So RRYY, so we are just supposed to multiply this, right? RR, RY and RY, then RY with this one, then RY with this one. So this is what we are getting in this first line. Again in the second line, small r, capital Y is multiplied with this with this. So this and combination we will be getting. So this first, this second, this third, and this fourth. So this all combination in this one also same. This is to be multiplied by this. So that subsequently is written here in this all four. This also with this. Okay, so what we are getting? We are getting 9 round seeds here. Okay, 3 yellow and wrinkle seeds here, 3 green and round seeds here, and one green and wrinkle seed here. So the phenotypic ratio in the F2 generation is 9:3:3:1. So the phenotype is this, and the genotype is much more complex. So that is not included in your 10th standard curriculum. In 12th standard, you will be studying this genetics in details. There I have already taught in a video about this in details. So if you want to learn more, you can refer to that video as well. So now we will again move back to the lesson where we were discussing about how to this sex determination. This process is going on with this inheritance of genes. So this. Now we have already spoken about this experiment here. So this experiment we have already spoken about. Now what we are telling, so as how do germ cells make a single set of genes from the normal 2 copies that all other cells in the body have? Now if the progeny plants inherited the single whole gene set from each parent, the experiment would not work. This is because 2 characters R and Y would be linked to each other, which cannot independently be inherited. So as per the scientists, it's told that they are R and Y. These are all linked to each other. So this is explained by the fact that each gene set is present not as a single long thread of DNA, but as separate independent pieces, each called chromosome. Okay, so they are not separate independent, we can say, but they are not in a single thread of DNA, but they are separate independent pieces called chromosome, and each cell will have 2 copies of each chromosome, each one from male and other one from the female parents will come. So chromosomes like this, one from the male will come, another from the female will come. So they are, this gene character is being set on any part of this chromosome, and they are linked. They are not independently coming. So every germ cell will take one chromosome from each pair, and these may be either maternal or of paternal origin. So one set of chromosome is in mother and one set of chromosome, suppose this is from the father. So when we are forming the germ cell, it will have one chromosome from mother and one chromosome from the father. So like this it will have, and these characters will be there on one segment of this. So they are linked. Okay, so they are linked. Is it clear to all of you? So these are linked. And we can say that such a mechanism of inheritance explains the result of Mendel's experiments and is used by all the sexually reproducing organisms. But asexually reproducing organisms also follow the similar rules of inheritance. Can we work out how their inheritance might work? So we will just see about what is happening there. That is, when two germ cells combine, they will restore the normal number of chromosomes in their progeny, ensuring the stability of the DNA of the species. Okay, so such a mechanism of inheritance explains the results of Mendel's experiment. So this video on sex determination I have done in details in one video, so I'll put the link down below. Please go to that lesson, that what determines the sex of a child, whether it is a boy, whether it will be a girl. So go to that part, and in details I have discussed that. So I'll not be discussing here because it will be making this video much bigger. So stay tuned with me, subscribe to my channel for more and more important and interesting topic as per your curriculum, and please do stay subscribed to my channel. Please do view my channel in full, don't skip in between, and if you're liking my channel, don't forget to share it with your friends. So thanks a lot for watching my videos.